Well, these are special moments for all of us. And Netra has done an absolutely fantastic job here. And, as some of you know, I've had a chance to restart my career here, this time on the West Coast. And a big part of that, and like we all build our careers, it's all really based on having great students, and in my case, staying out of the way. And I think this Netra really epitomizes that in many ways. She got her degree at the University of Toronto and came in with a lot of bioengineering experience and training and perspective, and has always been a very motivated and focused researcher, and also very collaborative and helpful to a lot of people. And I think really set the tenor for the reestablishment of a research group. And I think when I look at my group today, I think your fingerprints are all over our group regarding our culture. And I think it really set a great standard, and one I was hoping for, but it doesn't always happen. And I think Netra really set that stage for all of us. And so a lot of us owe you a great deal of gratitude. And we're excited about the direction you're taking professionally, if you get through this today. So with that, let me turn it over to Netra. Welcome.
Thank you, Joe, and thank you everyone for being here in person or on Zoom. It's my pleasure to be here today to share with you what I've been working on in the DeSimone lab over the past few years with my project titled High Resolution 3D Printing of Microarray Patches for Transdermal Applications. Now, I'm sure we're all familiar with needles. They're a small shot, but they offer a really big shield, particularly in the context of vaccines. With the increase in global vaccination campaigns and more widespread vaccination, several deaths for tragic diseases have been averted. But also in the context of blood tests and early diagnostics, being able to detect diseases early via a blood test has also improved survival rates, particularly in diseases like cancer. But the reality is, not everyone agrees with the word small. In fact, a lot of people have a very visceral reaction to injections. Some people experience dizziness, they experience panic, and that results in vaccine hesitancy and blood test hesitancy. And this is actually a disorder called trypanophobia, which is a fear of needles. And this affects 2 in 3 children and 1 in 10 adults. So we started thinking, what if you could design needles that went into the skin and not past the skin? Or in other words, micro-sized shots. And in fact, the skin is actually a really ideal candidate for drug delivery because it has a higher population of migratory immune cells, which are the cells you want to target for vaccinations. Your skin is also part of your mucosal system, so you can induce mucosal immunity, which results in the production of very specific antibodies that can help protect you from further infection. There's also a lack of sensory nerves in the skin. We only go about into the dermal layer here. So that means this process can be minimally invasive. And another advantage of intradermal delivery, particularly with what we call a microarray patch, is the fact that they can be self-administrable. So a microarray patch, or a MAP, consists of micron-sized projections that penetrate into the skin. And usually these tend to be very small patches with about 50 to 100 needles, and they allow you to painlessly administer cargo into the skin. So today, I'll be talking to you about how my PhD thesis has revolved around designing these new MAP platforms for transdermal applications. We'll start off by talking about what transdermal applications are and a novel framework that we've developed for classifying these microarray patches. Then we'll delve into the new MAP geometries that are enabled by a 3D printing technique developed in the DeSimone lab. Finally, I'll talk about latticed microarray patches, or LMAPs, which is kind of the central aspect of my PhD thesis, and how we can use this platform for various clinical applications. And finally, we'll take a little bit of a detour and talk about how we can deliver cargo into the skin, but also extract fluid from the skin for diagnostic applications, specifically looking at extracting interstitial fluid with MAPs. So my thesis kind of spans this realm of delivery, but also sampling. And hopefully I can show you the span of that today in my talk. So starting off with transdermal applications and a novel framework for MAPs. So as it stands, there are actually quite a few FDA-approved transdermal products. Some of them are administered intradermally, so a trained clinician has to actually put the syringe into your skin, go in at a very specific angle. Doesn't happen right all the time, but with this intradermal delivery, you can actually get an injection that delivers things like the influenza vaccine into your skin. There are also transdermal films, which people might be a little more familiar with, including things like nicotine patches. And there's also transdermal patches, which are a little bit different from the films because they are often made with an opaque material. And these can be used to deliver drugs that often combat motion sickness. And there are some microneedle products, but often these are used in the cosmetic setting and they're coupled with a syringe or some kind of injector. But what you'll notice here is, what about microarray patches? Why aren't there any FDA-approved microarray patches? Well, in order for MAPS to be a clinically relevant tool that's disseminated to people across the world, there are some optimal characteristics that they need to fulfill. These include something such as the ability to be easily applied and self-administrable, being pain-free, having mechanical strength to penetrate skin, and some other criteria are outlined here. And MAPs have been around for quite a while, since as early as the 1970s. So this got us thinking, how can we classify where MAPs have been and where we're heading? And this resulted in us creating a novel framework for MAPs based on the methods of making these MAPs. So in the 1970s, you can start off with first generation MAPs or Gen 1.0 MAPs. And a lot of these use manufacturing techniques from the microelectronics industry, such as techniques used to make micron chips and things like that. So because of this, the kind of geometries that you could achieve are very limited. They're all uniform needles, often made of materials like silicon. But in the early 2000s, they took a step further into Gen 2.0 MAPs, where now you can integrate molding to create similarly uniform geometries, but now the materials that could be used are expanded a little bit more. However, these materials still have to be moldable because they have to fit into the mold and harden, have enough strength to penetrate the skin. Where our lab really likes to play is in this realm of Gen 3.0 MAPs, where now you can use direct fabrication with techniques like 3D printing to create more complex geometries that have never been made before. And our lab has extended this even further to create Gen 4.0 MAPs, where you can tweak some of the 3D printing techniques that we're using to create these more complex map geometries that have channels in the back for added functionality. So if we take a little step back and look at what kinds of maps are already out there, if we look at some of the Gen 1.0 maps, again, the needle geometries are pretty limited. To make these maps is a multi-step and multi-day process because you have to change the mold every time you want to change the design. And some of these needles are also very brittle and experience mechanical failure upon insertion into the skin. So here I've highlighted from the optimal criteria that we talked about what's fulfilled and what might not be addressed by the Gen 1.0 maps. Moving into Gen 2.0 maps, again, we still have limited needle geometries. A lot of them are conical needles or square pyramid needles. You don't really see any cutouts within the needles. And this also requires a multi-step fabrication process. But now, again, we've expanded the scope of materials. More biocompatible materials that are moldable can be used with this process. The molding process also has its limitations in that often these needles have poor tip resolution and sharpness, which affects how they're inserted into the skin. And there are some Gen 3.0 maps that use different 3D printing techniques to make microarray patches, but these have still been focused on simple conical needle geometries. And a lot of the 3D printing techniques that are listed here that are used to make these maps take a lot of time. So to create these high resolution, small micron scale structures, it takes a longer time to fabricate the needles, which kind of impacts the ability to scale up and manufacture patches, again, to be disseminated across the globe. So that brings me to a key technique in our lab that was developed and allows us to create new map geometries that have never been seen before. And this technique is called continuous liquid interface production, or CLIP. And we've even been able to create a high resolution version of this technique so that we can resolve much finer features with 3D printing. Essentially, how this technique works is it's a software-controlled chemical reaction. So you have a 3D printed part or a file that you have designed. You can upload that into the printer. The printer will split that part into little sections and allow you to get your 3D printed object within a matter of minutes. And really, the key technique here is being able to use a photopolymerizable resin and having a window that's permeable to oxygen that's around us. Oxygen is an inhibitor of the polymerization reaction that the printer uses, and this allows you to continuously print a part in a rapid fashion. And the high-res aspect of this printer comes from the optics of the system, which is down here. We were able to find a lens, and a postdoc in our lab, Dr. Kaiwen Shao, who's now a professor at Texas A&M, I believe, she was a pioneer in creating this high-res clip technology. And what was really exciting for us is, since we're interested in using this in a medical application, there is a resin called Keysmut Hard, which has already had a file at the FDA since it's been used in Invisalign-like dental products. And this resin is compatible with the clip process, meaning we can have a biocompatible material that can be used to make these patches for delivery or sampling from human skin. So with this technique, we've been able to explore new classes of microarray patches that haven't been seen before. The first is a concept that we came up with in one of my early papers in the lab called dynamic maps or DMAPs. These have living hinge mechanisms embedded in the needles, which allows the needle to interact with the skin as it's being inserted or taken out. And this can help with keeping the needle retained in the skin to improve adhesion, which can also affect the ability of cargo to be delivered into the skin more efficiently. Another class that I would like to introduce here are the latticed microarray patches, or LMAPs. So these are the first generation of designs, and you can already see how we're able to gain complexity in the needle geometries that have been achieved by using high-res clip. So really, we're adding two new design variables here: the strut thickness, which is how thick the struts of the lattice are, and the cell size, which is how much void space you have in the needle to be able to trap cargo. And these are some of the first prints that we were able to make with these Gen 1.0 LMAPs, and I'll be talking a little bit more about the processes we use and what we've been able to do with these designs. Finally, I also talked about the ability to use induced channel backings into the needle to create these new microfluidic back maps, or MMaps. And this paper actually just came out in a preprint with some of our colleagues in the lab. And we've been able to integrate injection into that clip process so that we can create these free-form negative space structures. And we've even extended this to create a version that's integrated with the lattice map design that I showed you earlier, and it incorporates a checkerboard reservoir so that we can limit the amount of wastage that we have as we're delivering drugs into the skin. So to summarize here, hopefully I've been able to show you all the new classes of maps that have been enabled by high-res clip, and specifically the design flexibility that 3D printing offers, which enables us to iterate and create these new needle geometries. With high-res clip, we've been able to design needles that have compliant mechanisms that can interact with the skin during map insertion. We've been able to produce free-form negative spaces and channels that add functionality to maps. And we've also been able to create unmoldable needles, which I will now discuss how this platform can be used for versatile and tunable delivery. So that brings us to the LMAP, or the lattice microarray patch platform. And again, an advantage of using 3D printing is the ability to iterate. And because of that, the LMAP design has gone through a series of evolutions throughout my PhD. Starting off with the first generation LMAPs, again, the key variables that were governing design of this was the introduction of strut thickness and cell size. I was able to create a library of 150 different LMAP designs, which is shown over here, varying parameters such as the lattice shape, the properties of the lattice, and a lot of these needles were designed using a tool called N-topology, as well as a 3D printing tool developed by Carbon called the Design Engine. With these first generation LMAPs, we actually used a prototype high-res clip printer that was developed in the lab, and we were able to demonstrate that these needles are strong enough to penetrate skin, but there was some deformation throughout the process once you took the needles out of the skin. We were also able to show that these needles had much higher cargo loading compared to their solid map counterparts, which are some of those Gen 2.0 designs that I showed earlier, where it's just a solid needle with cargo on top. However, some of the challenges that we faced with this first generation of LMAPs was that it took weeks to optimize prints, just given the prototype nature of the printer that we had. So there was a lot of iteration involved in optimizing some of the needle geometries we were trying to achieve. There was also the requirement to add a non-biocompatible UV absorber into the resin so that we'd be able to resolve these fine features. And finally, the prototype printer had a very small build area. So if we wanted to make a patch with, say, 100 needles, we had to tile together a bunch of smaller patches. And we learned very quickly that glue is water soluble. So sometimes when you put these patches in water, they just discombobulate. However, really a blessing that came during my PhD was the ability to get an industrialized version of the 3D printer in collaboration with Carbon, which is a company my advisor founded. And having this industrialized version really sped up the ability to iterate and the kinds of geometries that we could achieve. And specifically, this is because Carbon actually created a custom vat design that has channels built into the reservoir, which helps improve oxygen flow and makes the prints a lot more reliable and reproducible. A lot of the needles in the Gen 3.1 category were created using a CAD software, computer-aided design software, called Fusion 360. So here we started with a library of about 12 different designs, and we were able to show that compared to the first generation LMAP, the LMAPs printed on this new printer actually had much higher strength compared to the first generation. And because Carbon was able to fix the optics of the system, we no longer required that non-biocompatible UV absorber, which meant we were able to make these needles and they'd be ready to go into humans immediately. However, we did observe that because of the design of the needles, there was some cargo leakage out of the skin. So if you have these latticed voids that are present on top of the skin, the cargo can actually leak out when it's being inserted. So this was when we turned to literature and found that you can actually add pillars to the needles to create a plugging effect so that the needles and the voids remain within the skin to reduce cargo leakage out of the skin. So this led to the final LMAP geometry that we focused in on, the Gen 3.2 LMAP, where we were able to create a library of 43 different LMAP designs that had a pillar integrated to keep the needle resident in the skin. So these are the 43 different designs that we were able to create. We varied certain variables for this needle design, including the number of lattice tiers in the needle, the shape of the base of the needle, since we saw in literature that the shape of the base can affect insertion into the skin. And finally, a key variable that was introduced here is the tapered strut. So what we saw before is if you have a smaller void volume, in order to increase your mechanical integrity, you have to put a lot more lattice struts. But of course, that means there's less cargo that you can load in your needle. So this tapered strut actually was a very unique way for us to get around that variable, because now you have a tapering in the needle strut that provides mechanical integrity, but it doesn't sacrifice void volume. So you can still load the amount of cargo that is desired to you. So looking at this, we were able to create 43 different LMAP designs, and we wanted to narrow down those designs to see what kinds of release profiles could be achieved. So the first filter that we employed was in silico modeling, or modeling the void volume and the strut surface area in the computer. And essentially that would allow us to get a series of different needle geometries that had varying surface area to volume ratio. And we hypothesized that this would affect how quickly cargo leaves the needle. And we wanted to be able to show that we could achieve tunable release properties with the LMAP. So once we did that, we narrowed it down to 15 different designs that had varying surface area to volume ratio. And we used finite element analysis to model the strength of these needles, since you wanted to make sure that the needles would be strong enough to penetrate the skin. And that resulted in 4 optimal needle designs. And those are the needle designs here. So there is a square base needle with 2 lattice tiers, a square base needle with 3 lattice tiers, a pentagonal needle with 2 tiers, and finally the standard solid needle, which is what we will be comparing to as a control. So all of these geometries had the highest mechanical integrity from in silico modeling, and they had diverse surface area to volume ratios, which we hypothesized would allow us to achieve differing release kinetics. So to start off, we wanted to make sure again that the needles were strong enough to penetrate the skin, and this meant performing mechanical characterization. So we did what's called instron testing, where you have a force plate that comes down on the needles, and you can measure the precise force at which your needles fail. So what we were able to see is that all of the LMAP designs are strong enough to penetrate skin, since it's approximately 20 newtons of force needed to penetrate skin. And of course, the solid needles were much stronger because they're an entirely solid body. We were also able to show that integration of a spring-loaded commercial applicator allowed us to get more precise puncturing of the skin because you had control of the forces that you were applying to push the patches into the skin. So together, these results suggested that LMAPs are strong enough to pierce skin and they do not deform upon piercing, which was key for us to see since some of the earlier geometries did have that deformation. We were also able to look at penetration across a patch to see how variable the penetration depths were for each needle. And what was really interesting for us is if you look at this heat map here, the LMAP actually has much more even penetration across the patch, whereas the solid maps, the central needles don't penetrate as deep. And this is also visible in the histology here where you can see variable penetration depths, but in the LMAP, it's more consistent. So this is because with solid needles, you actually have what's called a bed of nails effect. And if you've heard of, a medieval bed of nails, it's the same principle. Pressure fields overlap, and because of that, the needles actually don't puncture the skin. They just end up kind of deforming the skin. And we hypothesized that a big reason why the LMAPs are able to overcome this is because they have less surface area in contact with the skin. So that would reduce some of the friction forces that the needles experience. We were also able to visualize this in a video. And in the video down here, you see a solid map. And when finger pressure is removed, you see that the needle actually pops out of the skin. And that's because of some of these friction forces. Whereas in the LMAP, the needle stays resident in the skin. So that was great for us to verify visually. The other thing we wanted to achieve with LMAPs is demonstrating that this is a platform tool, which is agnostic to the cargo that could be delivered. So we wanted to show that the same needle patch could be used to deliver solid state cargo in the form of films. And we did this by looking at small molecule cargo in ex vivo pig skin and looking at solid state protein delivery in both pig skin and in rats. But we also wanted to show that you could use the same needles to deliver liquid cargo, which has never been done before, a single map platform that can do both solid state and liquid delivery. For liquid delivery, we chose small molecule cargo as well, and also nucleic acids. So things like mRNA, which I think we're all familiar with the COVID vaccines that we've been very grateful to have. So first, looking at small molecule cargo loading. We used a dye called methylene blue, which is a small molecule commonly used in transdermal applications. And we were able to show that just looking at loading of the dye, only latticed maps can trap liquid cargo. You can see that in the solid map, there's nothing because there are no voids that are capable of trapping the liquid cargo. So in the context of liquid small molecule loading, we were able to achieve 4 times higher loading in LMAPs compared to the solid system. Looking at solid state coating, now we actually incorporated viscosity enhancers that makes the liquid thicker so that it can coat the solid needle. So because of that, we now have some drug or some molecule cargo being loaded on the solid needles, but the LMAPs still have a larger void volume and surface area. So we were able to achieve 1.5 times higher loading compared to the solid maps. Next, we wanted to look at release kinetics because a hypothesis that we had is the varying surface area to volume ratio would affect how fast cargo releases out of these needles. So we started off by looking at release kinetics in excised pig skin, which is a great surrogate for human skin, just given the morphology of the 2 skin models. And we were able to show that the LMAPs did have differing release profiles, with the square 2-tier releasing cargo the fastest. So this is cargo normalized to the amount that you can load on the needle. And if you just look at the absolute amount of cargo delivered, the difference is even more stark. You can clearly see that there are differing release profiles with these needles. With solid state kinetics, it's a little bit harder to tease apart, but we still saw differing release kinetics, particularly at the 5-minute time point. And what this showed us is between the square 2-tier geometry and the pent 2-tier geometry, the most difference was observed in release kinetics. So both of these needles have differing release profiles. And that led us to think, what if we could create a hybrid patch where we combine those 2 needle geometries to have differing release profiles on a single patch? So here the cargo that we used was ovalbumin, which is an egg white protein that's commonly used as a vaccine antigen or a vaccine surrogate. And we were able to make a solid state formulation of this where you can dip coat the map and air dry it. So you have
reach out to all 5 of them and see who's going to respond.
Yeah. Okay, I think we're good. Awesome, thank you. So we were able to use ovalbumin, which is an egg white protein, and we essentially dip coat our maps and air dry the formulation so that we can create a solid state film that would be more thermostable and would not necessarily require being refrigerated or being part of the cold chain process. So to start off, we wanted to show that we could also load more OVA protein in our needles compared to the other Gen 1.0 and 2.0 needles that are out there. So these 2 bars here are the OVA protein that we can load on the LMAP. And you can see that we're able to have a much higher loading capacity compared to what dissolvable and coated maps that are already out there were able to achieve. Then we moved on to generate these hybrid LMAPs using the square 2-tier and pent 2-tier geometry. We coated the square 2-tier preferentially with an ovalbumin protein that was labeled with Texas red, which is a red fluorophore. And we coated the pentagonal ones with ovalbumin that was labeled with Alexa fluor 488, which is a green fluorophore. So we could essentially track release of these fluorophores out of the needles and observe the release kinetics that we're able to achieve. In a rat model, we were very excited to see that, as we observed in pig skin as well, the square 2-tier appeared to release cargo faster. And you can kind of see that in this plateau here. It reaches a much higher plateau at a faster rate compared to the pent 2-tier design. So what this suggested to us is you can create hybrid patches that have differing release profiles on a single patch by tuning the formulation as well as the LMAP geometry that you're using. Finally, for liquid state delivery, we chose mRNA as our delivery platform. And mRNA is very unstable by itself because there are a lot of enzymes in your body that can eat it up. So you need to protect it in a nanoparticle cargo, which is what they do for the COVID vaccine as well. So we used an MC3 lipid nanoparticle encapsulated with mRNA that encodes for firefly luciferase. This is actually a protein that's made by fireflies, and when it encounters a substrate, it luminesces. So you can detect that luminescence signal, and that's a measure of how efficiently your mRNA has delivered and if your cells have been able to take up that mRNA and make the target protein. So again, we started off first by looking at mRNA loading, and we were able to show that the LMAPs have more efficient RNA loading compared to other map systems out there. We also used an ex vivo pig skin model, but this time we kept the skin alive after you remove it off of the animal, and you can do this by keeping it alive in cell culture media, so that we could deliver our RNA cargo via LMAPs and show that the target protein is being produced. So the redder it is here, the higher protein expression that we have from the mRNA that was delivered. And we were able to see protein expression in as short as 1 minute application time, which is exciting for us because you want to be able to deliver this cargo quickly, especially, if you're looking at pediatric populations where you might not have as much time to keep the patch on the infant. We were also excited to see a dose-dependent response, where if we increase the amount of RNA that we deliver, we get higher protein expression. So this is great to see in ex vivo pig skin, but we also wanted to demonstrate this in vivo. So again, we looked at a rat model, and we were able to apply the LMAPs for different durations of application, and we were able to show very local expression of RNA, which was exciting to us again, because it shows that we're delivering to cells in the skin, which are then able to make the target protein. Finally, we also were able to collaborate with a company called 10Bio that actually gets excised human skin, and you can kind of tension the skin and keep it alive outside of the body. And we were able to deliver RNA again with the LMAP platform or with an ID injection, which is the other way of delivering cargo into the skin. And we were excited to see that LMAPs can transfect live human skin to a similar level compared to an ID injection. So to summarize, LMAPs are a platform that's capable of increased cargo loading and improved skin penetration. We were able to show that these needles are able to spread the pressure a lot better so that you have more even penetration across the patch, and that they stay adherent in the skin, whereas solid maps get popped out. We were also able to show that LMAPs are a platform for both solid state and liquid state cargo delivery, and we were able to show this in the context of 3 different cargo types: small molecules, proteins, and nucleic acids. Finally, we were also able to show that cargo release kinetics can be tuned by altering the LMAP geometry as well as the excipient formulation. And you can even integrate these different geometries to create modular LMAPs that have differing release profiles on a single patch. So putting all of this together, this actually sets the stage for using this technology in the context of transdermal vaccination, which is what I will be talking about next, clinical applications of LMAP technology. So if you recall, when I showed you the slide earlier about the advantages of transdermal drug delivery, one of the advantages is that the skin has more immune cells. And of course, this makes sense since it's your first line of defense and there are a lot of immune cells floating around looking to see what foreign pathogens are there so that they can prevent them from infecting you. However, vaccines also act on immune cells. And in fact, they work to educate the immune system by gently introducing the enemy, in this case, the pathogen, to your immune system. And the way they do this is signatures of the pathogen called antigens are introduced into the body. And when immune cells encounter these antigens, they make antibodies, which protect you and can recognize that antigen in the future. So when a person is reinfected, the antibodies can then be used to neutralize that pathogen so that you don't have as strong a response to the infection. So putting this together, LMAPs could be a vaccine delivery platform. And given that we're also able to deliver different states of cargo, both solid state and liquid state, we wanted to see if we could use LMAPs for different vaccine applications. First, we were interested in looking at liquid state COVID RNA delivery. And again, I will delve into the formulation here, but similarly, we have RNA that encodes for a part of the SARS-CoV-2 virus encapsulated in a lipid nanoparticle, which we can then load on the LMAPs and deliver into the skin. But we're also very fortunate to be working with the Bill and Melinda Gates Foundation on using this platform for liquid and solid state delivery of measles live attenuated viruses as a form of measles vaccination. To start off, I'll talk about liquid state delivery of SARS-CoV-2 RNA. So the formulation that we were able to obtain to work on integrating with the LMAP platform was graciously supplied by VLP Therapeutics, which is a company that we're fortunate to work with here at the DeSimone Lab. And essentially, they have a very unique RNA formulation that encodes for the receptor binding domain of the SARS-CoV-2 virus. So this is a part of the spike protein of the virus that actually allows the virus to attach to cells and infect cells. So they've been able to create a messenger RNA version or mRNA version, and this is specifically for the gamma variant of SARS-CoV-2. And essentially how mRNA works is you have a single strand of RNA, and that usually makes a single protein. But VLP Therapeutics is unique in that they were able to create a self-amplifying RNA or saRNA, where there is an amplicon or an amplifying motif embedded in the RNA, which allows you to replicate the RNA and create multiple proteins from a single strand. So this got us thinking, if we were able to integrate saRNA with the LMAP, maybe we could reduce the dose of the dose of RNA that's being delivered. And as I said earlier, the RNA needs to be protected. So the lipid encapsulation system we used here is from a company called Genvoy, and they have a standard lipid mix that can be integrated with RNA. So we used the Genvoy lipid nanoparticle. So to start off, we wanted to evaluate this formulation in vivo, and we were really fortunate to collaborate with VRI, a veterinary research institute in Ames, Iowa, and we were able to test these formulations in live pigs. So to start off, we looked at the saRNA Genvoy lipid nanoparticle, the mRNA Genvoy lipid nanoparticle, and a placebo that was just a saline injection. We looked at delivering 10 micrograms of RNA, either intramuscularly, so into the muscle of the animal, or intradermally via that perfectly positioned intradermal injection. We were very excited to see that after the prime dose, which happened on day 0, we did see elevated SARS-CoV-2 specific antibodies in the animals for all of the different applications and all of the different RNAs that we tested. And if we look at the boost dose, which was given on day 42, we see an even higher SARS-CoV-2 specific antibody titer. So to us, this suggests that the formulation works and we're able to induce COVID specific antibodies by delivering this RNA. And I was really fortunate to go out to Ames and actually see how a lot of these injections are performed. And that's me with Wilburina, a friend that I made in Iowa. So now that we are able to show that the injections work in animal models, we wanted to now integrate that formulation with the LMAP. And for this, we used a mouse model. So we were looking at vaccinating mice with RBD RNA that is on the LMAP system. However, mouse skin is very different from pig skin or human skin or rat skin. So we very quickly realized that we need to redesign the LMAP system to be able to deliver into mice. And again, this is the advantage of 3D printing. We can recognize the variables that impact the efficacy of our design, and we can quickly iterate on that. So what we did is, instead of going with the 2-tier geometry, we created a 1-tier needle geometry that would put the opening closer to the tip of the needle. And we increased the spacing of the needles on the patch. So we had 1.6 millimeter spacing, which would allow us to, again, get better penetration in the very soft, thin mouse skin. So this is the 1-tier LMAP that we decided to move forward with. We loaded liquid SARS-CoV-2 saRNA embedded in the Genvoy lipid nanoparticle onto these LMAPs. And we did a first dose of vaccine at day 0 and did a boost dose at week 5. And I'm very thankful to our collaborators at UNC Chapel Hill who were able to perform these studies for us and were critical in getting this project going. So if we look at the antibody titers week 4 after that first dose, we can see that we are already able to create some SARS-CoV-2 specific antibodies. And what we did observe, however, is when we compare it to the ID and IM controls, the magnitude was about 1 order of magnitude lower. So we still had a little bit of room to create a robust antibody response. However, when we did that second boost dose, we were actually able to see an increase in antibody titers, and we were now much closer to the same order of magnitude that the intradermal and intramuscular controls had. So to us, that suggested that the LMAP could be used for RNA delivery, but what about a different type of vaccine or a live attenuated virus vaccine? So measles actually uses a live attenuated virus, which is a weakened measles virus that will induce an immune response, but not infect a person. And this is actually being used to immunize children across the world right now against measles. And in fact, even the World Health Organization recognized that MAPS could be a good platform for measles delivery, and they created a target product profile for a microarray patch that would deliver the measles virus to create a measles vaccine. Some of the criteria that they had is being able to deliver the cargo in less than 5 minutes, again, because the target population is infants. However, less than 1 minute would be even more ideal. They also wanted us to demonstrate equivalent response to an intradermal or subcutaneous injection control. And finally, they wanted this formulation to be stable at 40 degrees Celsius for 3 days, because this is going to be transported into low-income environments where they might not have access to cold chain. So keeping all of this criteria in mind, we started to design a liquid version of the LMAP platform where we loaded liquid state measles live attenuated virus onto the patches. And we added some excipients such as gelatin and sucrose that would allow us to stabilize the virus. And we were able to see very robust responses with 1 minute and 5 minute applications compared to an intradermal injection. So seeing this data, we were able to achieve the deliver in 5 minutes or 1 minute criteria. We were able to achieve equivalent response to an intradermal or subcutaneous injection control, but we still need to work on this 40 degrees Celsius for 3 days element. So there are colleagues in the lab who are going to be taking this work forward. And more recently, we were actually able to take a stab at this and got very preliminary data that we are able to keep it stable at 40 degrees Celsius, which was exciting to see. So to summarize here, LMAPs can be used as a platform for transdermal vaccine delivery. And we've demonstrated this through the generation of robust antigen-specific antibody titers in mouse models, specifically looking at liquid state delivery of SARS-CoV-2 encoding saRNA and live attenuated measles viruses. So together, we were able to show that we can deliver the liquid state version of this cargo, but in order to create a platform where you can deploy these vaccines in a more widespread manner, you need to create thermostable MAP products. And for that, we have colleagues in the lab who are focused on lyophilizing or air drying the cargo. And we're hoping that this will be a promising strategy to keep our sensitive cargo stable in the MAPs. So thus far, I've shown you how 3D printed MAPs can be used to deliver cargo. But remember, I also said we can use this platform to sample from the skin for diagnostic applications. So now I'd like to talk about some of the 3D printed platforms we've been working on to collect interstitial fluid for diagnostic applications. So dermal interstitial fluid, or ISF, is a great alternative to blood. In fact, we have 3 times more ISF in our body than blood, and ISF is in constant exchange with blood because of all the capillaries in your skin. So this suggests to us that ISF has a similar composition to blood. It's a lot easier to collect with transdermal strategies, and we can also collect this in a minimally invasive and self-administrable manner with MAPs. And in fact, map-based ISF collection is gaining a lot of traction now, and there are some groups who are focused on collecting ISF using different techniques. Some of them use this kind of pressure ring method and have a capillary tube attached to the needle. Others use a poke and pull method where they'll puncture the skin and then use a vacuum to create blisters that draw the ISF out of the skin. Some of them also use a combination of these methods, but the key drawback that we've seen across the board is that the collection rate is very slow. So in order to collect a small volume, like 1 microliter in this case, it takes 5 minutes. So in order to make this fluid useful for diagnostic applications, we really need better methods of collecting ISF in a more robust manner. So with a colleague, Andy, in our lab, we started thinking that ISF in skin is a lot like groundwater in soil. It's kind of these water droplets that are trapped in a matrix, and you have to be able to draw the fluid out of that matrix. So we turned to Darcy's law, which is a law that relates the flow rate of a fluid in a porous medium to the pressure that's applied. So it became clear to us that we needed to create a pressure gradient that would allow us to create flow of ISF and draw it into our device so that we could collect it. So for this, we designed a 3-part ISF map device where you have a microarray patch that fits into this collection plate with holes so that you can puncture the skin and you remove the microneedles. And then you create pressure by applying a large pressure block so that there's pressure applied around each of the little openings. And finally, the ISF that's collected can be stored in this collection plate and spun out for further analysis. So using this device, again, because we're using a material like key splint hard, we're able to translate this and test it in humans. So one of the highlights of my PhD was being able to work with Andy to actually evaluate efficacy of this device in human volunteers. So we had a small IRB oversighted study where we were able to test this device in 10 human subjects. And we saw variable ISF collection, but what was exciting for us is that the puncture sites that are generated from the device heal within 24 hours. And what was most exciting also is that the process was relatively painless, with most people rating it a 1 out of 10 on the scale over here. More interestingly, we also saw a trend in the amount of ISF collected with the age of our participants. So the older participants were, the more ISF we were able to collect from them. And a hypothesis that we had is because as you age, your skin conductance increases. All of the elastin and collagen in your skin gets a little bit more open in terms of the network design. So that could allow fluid to flow a lot better, enabling higher ISF collection. And with our work, when we compared it to the other map-based ISF collection methods, we're 1 to 3 times higher in terms of the collection rates that we can achieve. So again, pointing to being able to create more robust methods for ISF collection that can allow us to learn more about ISF, but also collect ISF for diagnostic applications. We wanted to delve into the clinical utility of dermal ISF. So in 3 of our patients, we were actually able to compare the proteins that we isolated from ISF to the proteins that we found in their blood through a finger prick blood collection. And we were able to see that the most abundant proteins in both ISF and plasma consistently compose of albumin, immunoglobulins, and apolipoproteins. And immunoglobulins are actually part of the antibody structures that provide immunity. So this also got us really excited because it suggests that ISF could be used for immune monitoring and to see how people respond to vaccines. We were also able to cross-reference the proteins that we ID'd with the National Cancer Institute's Early Detection Research Network biomarker database, and we found that about 149 of the proteins we ID'd are also found in the NCI database, and Most of these are correlated with biomarkers for breast cancer and ovarian cancer. So that was also exciting to see in terms of what kinds of applications we could use dermal ISF for. Now, relating this back to the LMAP design, given that the LMAP has void structures in it, I started thinking, what if we could use this to not only deliver cargo into the skin, but also to draw ISF out of the skin? So I adapted the square two-tier LMAP design, which had the most robust delivery properties and mechanical properties. And I essentially adapted the design to have channels along the back so that we could draw fluid up out of the needles, integrated that with the reservoir so that you could get a monolithic design where you sandwich an absorbent material in between these two components, collect ISF, and then you can do on-chip dilution to release the ISF from the device. So comparing the LMAP to the three-part ISF collection device that I just introduced, it was very interesting to see because a three-part device uses solid needles. Again, there was higher penetration depth observed with the LMAPs compared to the solid needles in the three-part device. And we were also able to see that when we compare ear skin to back skin, pig back skin is very thick, pig ear skin is a little thinner. The penetration depth plays a huge role in the amount of ISF that you can collect. So if you penetrate deeper into the skin, you can collect more ISF. So this led us to think, oh, maybe the LMAP system, given its improved penetration, could allow us to get higher amounts of ISF. So when we tested this in excised porcine skin, we were actually able to show that the LMAP device collects about 2 times more ISF from this porcine skin compared to the three-part device that I introduced. And you can actually see what the ISF looks like. So you'll see it's a little red here, and that's because the ears were freshly harvested, so they still had a little bit of blood contamination there. But the ISF gets trapped in the absorbent material within this LMAP device, and you can actually centrifuge it out to force the liquid out, and you get a clean device again. All of the ISF gets spun out. So this is also an easy method to be able to collect ISF from this device. We also looked at the concentration of proteins between the two collection methods, and we saw that they were very similar, with the LMAP device having a slightly higher protein concentration compared to what we saw with the three-part device. So to summarize here, I've shown how high-res clip can be used to create map-based devices for rapid dermal ISF sampling through the introduction of the three-part map device, as well as an LMAP-based ISF collection device. And with the preliminary screening that we've done in ISF, this really sets the stage for us to use ISF in different applications, including localized omic analysis. So we can see how genomic and transcriptomic composition varies across the body. We could use this for disease diagnosis and predictive assessment. Again, given the abundance of immunoglobulins, we could use this to see how people respond to therapies and vaccines and if they're generating antibodies as they should be. And finally, you can integrate these map systems with wearables as well for continuous monitoring. So that brings us to the conclusion today. So we started off talking about how high resolution clip enables the design of new Gen 3.0 and Gen 4.0 maps. And then I introduced the lattice map system, or the LMAP system, which is a new Gen 3.0 platform capable of solid and liquid cargo delivery, as well as capable of tunable release profiles on a single patch. Then we extended this technology to talk about how we can use LMAPs as a platform for transdermal vaccination, and that we're able to generate robust antigen-specific antibodies for both SARS-CoV-2 RNA delivery, as well as measles live attenuated virus delivery. And then finally, we took a little bit of a turn talking about how we can use maps for rapid dermal ISF sampling, and related this back to the LMAP work again by showing how that single device can be used both for drug delivery and for ISF sampling. So looking forward, really development of 3D printed map technologies sets the stage for more research in designing novel carriers for intradermal delivery, because a lot of the lipid nanoparticles, for example, are optimized for intravenous or intramuscular delivery. Now you're going at a more surface level, so maybe we could design carriers that are able to release cargo faster and more efficiently. With the LMAP system particularly, this also sets the stage for a platform that can integrate delivery and sampling on a single platform. And with the integration of 3D printing, now we also have an idea of what kinds of new 3D printing resin materials could be useful for these applications, particularly to expand the biocompatible materials that we can use, the mechanical properties that can be achieved, as well as improving resins for printability so we can make these devices faster. For further technology development, our lab is actually looking at precise cargo loading in these maps using a picoliter dispenser so that you know exactly how much volume is going into each needle. As I mentioned earlier, we're looking at lyophilization of cargo in maps to create more thermostable products. And we're also looking at designing new map applicators that are easier to use and would allow us to get that even force distribution throughout. So with that, I will now be moving on to acknowledgements and would like to thank all of the amazing people, both within the DeSimone Lab and beyond, who made this work possible, our collaborators, the leadership team in the DeSimone Lab, all of the administrative staff who did all of the things behind the scenes so that we could focus on research. I'm also very grateful for our funding agencies, the Knight Hennessy Scholars Program, as well as the Siebel Scholars Program and the Bill and Melinda Gates Foundation, and Welcome Leap. Of course, I also have to thank my oral committee who provided a lot of strategic feedback and guidance as I was navigating this process. First and foremost, my advisor, Joe, thank you for believing in me and for giving me opportunities that I couldn't even imagine. Jennifer, thank you so much for your guidance as well throughout committee meetings. And Steve Quake, who I believe is on Zoom, thank you for all of the lovely dialogue that we've had, very impromptu, on how we can use ISF. That's been really exciting to see. And of course, John, super grateful to have you here and have you be a part of the process. Thank you for chairing the committee today. And Catherine, thank you for all your valuable insight on the immunological aspects of this project. Aside from committee and lab members, I would also love to thank my friends, many of whom are in the room today, from my friends in the Knight Hennessy Scholars community to friends at Stanford Ragapella, which is an a cappella team that I was part of. Thank you all for making starting a PhD in the middle of a pandemic bearable and possible. Could not have done this without any of you. And of course, there is one very special friend who became family. And I have to cite Gaurav for his thesis defense because this is exactly how he introduced me and I don't want any plagiarism. So gonna cite you there. Seriously, Gaurav, thank you so much for being there every step of the way. I think you are easily the highlight of my PhD and one of the biggest achievements, if not the biggest achievement. I know it must have been hard the past few weeks. It's been stressful. There's been a lot going on, but there's no one else I would want by my side. And I'm looking forward to making lots of memories and sharing a life with you. I love you. Okay, before I cry, I also want to thank my family, many of whom are here today. My mother, Nisha, I remember when you took the first lab tour with me in high school and you were like, oh my gosh, this girl loves her research equipment. Who knew I would be here doing the PhD? Thank you for driving me for like 4 hours every week to my research internship. Could not have done this without you. I'm thankful to my grandparents. My grandma's here today and my grandpa is probably watching from a better place, but I'm very grateful to have you all, to have you having raised me. Could not have been here without you. My aunt, uncle, and cousins who are on Zoom today. And of course, the new family members that Gaurav brought into my life. I'm so grateful for all of you. I'm honored to be a part of this family. And I hope I did you proud. So with that, thank you all for being here and happy to take any questions.